Plant agriculture is vital for human welfare. We depend on plant crops to feed ourselves and our livestock. As our population grows, meeting those needs becomes increasingly challenging. According to the United Nations Food and Agriculture Organization, agricultural production needs to increase 70% to meet predicted population numbers by the year 2050. Yet, most land that can be used for farming is already being farmed. More than a third of the world's land surface is devoted to agriculture. This means increasing demand must be met by technology rather than acreage. Technology that can help crop plants overcome threats like pests, diseases, and severe weather can increase crop yield. Laboratories working on solutions to these kinds of problems are key to making that happen. The Clark Lab at the University of Utah is researching one such problem: spider mites. This tiny pest, no larger than a pinhead, can barely be seen without a microscope, yet they are extremely destructive. The Clark Lab is studying how genetic variation can help plants like maize fight off spider mites. Maize, known more popularly as corn, is the most widely grown crop in the United States. We produce 361 million tons of maize every year. Maize is important. It helps feed people and livestock. Products derived from maize are also used as fuels and to make industrial products. Spider mites are a problem for maize. Like their name might suggest, these tiny pests are arachnids. That is, they're more closely related to spiders and ticks than to insects. Spider mites even spin webs like spiders, but not to catch prey. The webs protect mites and their eggs from predators. Ticks use piercing mouthparts to suck blood from animals. Likewise, spider mites feed by sucking fluid out of plant cells. Leaves that have been damaged by spider mites have white patches after mites have sucked out the contents of plant cells. Even though mites are very small, they can inflict a lot of damage and can be hard to control. This is largely because of two factors. First, they can quickly develop resistance to pesticides. Some kinds of pesticides can even cause spider mites to reproduce more than usual. The other factor is that spider mites thrive in hot and dry conditions. Droughts can cause population explosions. As they reproduce, numbers can quickly grow to reach thousands to tens of thousands of mites on just single plants. Mite infestations during drought have caused maize yield losses as high as 47%. As we face new climate challenges, spider mites are becoming a growing agricultural threat for maize. People have been selectively breeding maize for thousands of years. Today, there are many varieties of maize. These maize variations have different traits. They differ in features like seed shape, color, and size. Most interesting for the Clark Lab, though, is that maize can vary in the chemical defenses it uses to deter pests like spider mites. This trait variation can mean life or death for a maize plant facing a serious mite infestation. The Clark Lab is working to identify the genetic differences responsible for these traits and figure out how they work. Working with spider mites is challenging because of their size. Researchers in the Clark Lab use a microscope to see the mites so they can collect them to use in experiments. Pipette tips plugged with cotton are attached to a vacuum. Researchers gently touch the tip to the tiny mites to suck them up. Ice slows the mites down so they don't spin webs inside the tips. In the greenhouse, different strains of maize are carefully infested with mites to see how they withstand an attack. To keep the mites on one section of the leaf, researchers apply a sticky substance to make a barrier. The mites are released onto the leaves of different maize varieties. Researchers can then compare their responses. After the mites have had a chance to feed on the leaf, researchers measure the amount of damage. In this experiment, the B73 maize line is visibly more damaged than the B96 maize line. Researchers collect the leaves and scan them with a computer program. The program calculates the amount of leaf that has been damaged. Researchers then sequence the genomes of the different maize varieties. They compare their genomes and pinpoint the genetic differences. These differences may explain why lines like B96 are not damaged by spider mites. Researchers also identify the protein products that the genes code for, and see when these genes are switched on and off in response to mites. By decoding the genetics of plant chemical defenses, the Clark Lab hopes to contribute to the development of maize varieties that are better able to defend themselves from spider mites. Technologies like these are vital for meeting our growing plant agricultural needs.